ladies and gentlemen, the books is in. The books are in. Yep. They in. Here. ETM Hotel. My bad, my bad. I was y'all seen my mic moving, huh? Yeah, share the show. We finna get into it right now, man. Give me a second. I'm going to start y'all off with a little conversation between a podcast from New England Journal of Medicine. And we're gonna get it cracking, man. We're gonna get it cracking. So y'all should be able to hear me pretty good. I'm gonna go ahead and get into it. Here we go. Listen to the questions. It's going down.
Michigan, introduce yourself by telling us your name and your title.
Sure. My name is Adam Loring. I'm an associate professor at the University of Michigan. I'm in the Division of Infectious Diseases and also in the Department of Microbiology and Immunology.
Could we start by having you tell us a little bit about coronaviruses and what they are and how they behave?
Sure. Coronaviruses are a group of RNA viruses. They're different from many RNA viruses in that they have much longer genomes, whereas many RNA viruses have genomes that are about 10,000 to 15,000 bases long. The coronaviruses are up to 30,000 bases long. So they have a lot more genes that they can encode, and they behave a little bit differently. They also have a lower mutation rate than most other RNA viruses, so they accumulate genetic changes more slowly. Like other RNA viruses and some DNA viruses, they also will recombine with each other, so they'll swap genetic material. And that was potentially important in the origin of SARS-CoV-2. And then, as we know from the pandemic and from prior experience, there's been spillover events with SARS and then MERS and now with SARS-CoV-2, where coronaviruses from animal species get into the human population, and in this case have caused a pandemic.
Do I have it right? Do coronaviruses cause the common cold?
Oh, yes. Thank you for bringing that up. So, yeah, there are 4 or 5 what are sometimes called common cold coronaviruses or seasonal coronaviruses, which fall into various genetic groups that have been with us for some time and that every year cause a number of cold or potentially flu-like illnesses in people across age groups.
I may be jumping ahead of myself here a little bit, but we now know that there's a couple of vaccines out there that seem to work extremely well against SARS-CoV-2, which begs the question, why haven't we had a vaccine against the common cold up to this point?
Sure. Well, the common cold, as we know it, is caused by many, many different viruses, not just the, quote, seasonal coronaviruses we just discussed, but rhinoviruses, parainfluenza, adenoviruses, a whole number of different viruses. Even within the family that causes the common cold, what most people experience as the common cold, the rhinoviruses, there are well over 100 different serotypes of rhinoviruses. So the diversity is not unlike what many of your listeners would be familiar with pneumococcus and the number of serotypes of pneumococcus, the causative agent of much of pneumonia. And so it's been very challenging to be able to design one or even a couple vaccines that would address that many different types of rhinovirus or the number of different viruses that can cause the common cold.
I bring it up because everybody's familiar with the common cold, and one feature of it is that when it's cold and flu season, everybody gets it, and there's almost nothing you can do, or seemingly almost nothing you can do to stop it. And so early on in the pandemic, when I was interviewing somebody and he mentioned that with time, everybody's eventually going to get CoVid19 because it's a new virus for which there's no immunity and it's just going to spread through the population. The only thing we can hope to do is either develop a vaccine or slow it down, but we won't be able to stop it. Is that a fair assessment? One way to maybe explain why this disease is going through the population the way it is that it's much like the common cold. Is that correct?
Well, I think there's probably a few reasons in there. That one is, of course, there's no prior immunity to SARS-CoV-2, so everyone's susceptible, whereas with the common cold, you can have, of course, different people who have experienced it with different intensities, and so there's going to be heterogeneity in the immunity. And of course, the other big difference is, as we've seen, some of our non-pharmaceutical interventions have slowed down the spread of SARS-CoV-2, and perhaps also slowed down the spread of some of the viruses that cause the common cold or flu. Masks, social distancing, the things that are generally reducing transmission of respiratory viruses will also tend to reduce the transmission of these, more common viruses that have been with us for some time.
I'd like to go over a few concepts that get talked about that many of our listeners may not be familiar with. So you mentioned earlier that these viruses tend to mutate, and once they do mutate, various things can happen. So I want to run down some terms that get used that people may not be familiar with, and maybe you can explain them in relation to SARS-CoV-2. One is the concept of natural selection. What is that, and how does that relate to what we're dealing with today with COVID-19?
Sure. So natural selection, we commonly think of it as survival of the fittest. That comes in two flavors. There's what's called positive selection, where there is, a genetic difference or an individual in a population that does something better, that's at a competitive advantage. So with positive selection, that individual or a mutation will increase in frequency in a population. And so an example for a virus would be a virus that is better able to bind its receptor or better able to transmit or maybe better at escaping our immune system. So it's going to have a competitive advantage, a leg up on the other viruses around it. And so it's going to be more fit in general and increase in frequency. Then the flip side is what's called negative selection. And that's if there's a mutation that makes the virus less good at doing something or worse at doing something. It's a truism that it's easier to mess something up than it is to get better at something. And so most mutations will tend to actually make the virus less fit. And so those mutations, for example, if the virus is not able to bind its receptor as well, or if it has a mutation that disrupts one of the functions of its other proteins such that it can't replicate or copy itself, that virus will, eventually disappear from the population. It will decrease in frequency, it'll lose out, and that's called negative selection. And so those are kind of how we think about the main forces that drive, mutations or viral variants up or down over time.
What's viral fitness?
So the classic definition is pretty nebulous. It means how someone, or in this case a virus, fits with its environment. And that's how Darwin initially thought about it. And so there are various definitions of fitness, even in virology, that we always need to be clear about. I think the purest definition in thinking about fitness these days is how well a virus reproduces, how many offspring it has. And so that can be anything from how well a virus is spreading from person to person, because it only can grow and copy itself by finding new hosts, or it can be something as simple as how well it makes copies of itself within an individual person. And so it's always important to be clear about kind of what scale you're talking about when you're talking about evolution of viruses. Are you talking about at the scale of a host and what's its fitness in a host? Or are you talking across broad swaths? And so how well a virus does at spreading across a country, which is another measure of fitness. And that might have things to do with how it does within a host, but also how it transmits from one host to another, or how it survives in the environment in between hosts. So those are all things that kind of integrate into the concept of viral fitness.
Another term that I've heard that I don't quite understand is founder effect.
Yeah, founder effects are very important, particularly for viruses. And so founder effect refers to when the genetic diversity or number of genetic differences or mutations in a population is reduced because the population itself has gone under some sort of reduction. And so then what happens is all the genes downstream, many generations, all the mutations are linked back to those initial founders. And so I think the best way to understand a founder effect is the analogy to Native American populations. So the gene pool of the Native American populations were derived from the founders who migrated over the Bering Land Bridge many years ago, and there weren't that many. And so it's kind of this subset of the genetic diversity that was around at that time. And then those founders established the population. And so the genes or mutations or variants that came with those individuals way back are carried on today. And they are not necessarily carried on because they were selected. So it was a chance event in terms of which individuals made it across. The key thing with founder effects is that you can get changes in the frequency of genetic variants or mutations due to chance, due to whoever was there at the beginning, whoever the founders were. In viruses, founder effects can really be very important. For example, if a person in an airplane happens to carry the virus over from another country, then whatever the gene sequence of that virus is going to be the founder of the population in the United States, for example. And so it might not be because the virus was any better at spreading in the United States. It might just be because it was the first one there. Another example of a founder effect in viruses is when you actually have transmission from one person to another. So one person might have a lot of different viral variants within them, but when there's transmission, maybe only one or two of them kind of make it across to the next person. And so there will be a founder effect. So all the viruses in that next person will only have one or two variants, even though there were more within the donor host. The recipient might not have as much diversity to start off the infection.
All right, so I wanted to stop that bill right there. If you were paying attention to anything, they had a lot of, he was asked a bunch of questions. Now, the person that was responding to the questions is from the New England Journal of Medicine. And he was just being questioned by the team. And they were just building on, the RNA-based, stuff that we're about to get into a little bit, but not necessarily what he was referring to, but more so what the mRNA vaccines are, how the SARS-CoV-2, came about. Y'all heard him referencing at the tail end of that. He was referencing, different variants, how variants occur, from one person to start and then it'll, kind of go on to another. He talked about natural selection. You know what I'm saying? All of these things that we're privy to, what some of us are privy to. However, let me go ahead and share my screen and let's have some fun real quick. I don't want to keep people too long. However, I do want to get this information out. So let me double check and make sure y'all can see my screen. Sorry about the audio earlier. All right, so it looks like I am showing on Facebook. On YouTube. Again, like the show, share the show. I appreciate everybody for tuning in. So we're gonna deal with what are mRNA vaccines and some other stuff, but I just kind of titled this "What are mRNA vaccines." By the way, peace to CK, Sister CK in the chat. If y'all don't know anything about it, if you click on her YouTube page, she actually got a video up where she talks about her experience with receiving the vaccine. She actually, received the Pfizer vaccine. She hasn't mutated, grown another arm, a leg, or got hair growing under her chin, or her DNA is not altered. She has not had any adverse effects, just the typical stuff that she was expecting. They gave her a fact check sheet and her vaccination card, so she is armed to present her information. She did say initially it did not hurt. She did have a fear of needles, but for this one, she said it did not seem to bother her whatsoever. That it was only a couple of hours later that she began to develop, some little soreness in her arm. Now, I haven't asked her about how she's feeling today. As far as the, related to adapting 24 hours after vaccination. But she has had a vaccine. So shout out to Sister CK. See you in the chat. And I know later on, I don't know if the pseudo killers will be live later on, but I'm pretty sure something will come about. So today we're just basically going to define the role of what mRNA is, pretty much just defining that role. What are mRNA vaccines, how they work, how effective mRNA vaccines are, then a brief look into the mRNA vaccine ingredients. Then we'll conclude. I know one big thing is, CK was like, yo, we need to get into the actual vaccine ingredients. Now, I have mentioned the ingredients before, brought them up on screen, but I didn't elaborate. And for people who are unfamiliar with it, we're actually going to demonstrate and show those. Now, all the sources that you'll see in this particular presentation at the end, all the sources are in the description. So if you're curious about anything presented in this particular bill, I gave them to you right there in the description of this particular video. One thing that I forgot to add is to go to CoVid19TV.com, Learning Center, purchase all of our books. Also go to Amazon.com, get Killmonger's Corner Volume 2. Also, make sure you go to FamilyConnectedApparel.com and purchase either your Niger Valley clothing, Mboka clothing, Marcy Ward Clan clothing, Kofi Paisai Research Team clothing, As I Learn, We All Learn clothing, and then another line of clothing coming soon. So please support. I'll drop the links to those in the chat. Appreciate everybody being a part of the bill today. All right, so the role of mRNA vaccines. Well, the first one is messenger. We all should know what it means. mRNA means messenger RNA. It carries the genetic information copied from the DNA in the form of a series of 3 base code words, each of which specifies a particular amino acid. All right, they use words as basically wordplay for that. If we're trying to communicate effectively. Now, the second role of mRNA is a transfer RNA, which is tRNA. It is the key to deciphering the code words in the mRNA. All right, each type of amino acid has its own type of tRNA, which binds it and carries it to the growing end of the polypeptide chain if the next code word of mRNA calls for it. Now, the correct tRNA with its attached amino acid is selected at each step because each specific tRNA molecule contains a 3-base sequence that can base pair with its complementary code word in the mRNA. Now, ribosomal RNA, all right, rRNA, associates with a set of proteins to form ribosomes. Now, these complex structures which physically move along an mRNA molecule catalyze the assembly of amino acids in the protein chain. They also bind tRNAs and various accessory molecules necessary for protein synthesis. Ribosomes are composed of a large and small subunit, each of which contains its own rRNA molecule or molecules. Now, we'll break it down a little bit later. I know I kind of lost a couple people with the tRNA, mRNA, and rRNA, but I don't know if y'all knew that there was a tRNA, mRNA. Well, you knew a mRNA as a messenger, which is basically the communication piece for what needs to happen to transpire inside of the body to tell it how to react or to protect itself against anything that's coming in that's similar to what it is. Now, what are mRNA vaccines? Many traditional vaccines put a weakened or inactivated germ or virus into your body to trigger an immune response. However, mRNA vaccines are different. Instead, they teach your cells how to make a small, harmless piece of the virus to trigger an immune response inside our bodies. So when Sister CK got her vaccination yesterday, what is transpiring over the next few days, and I'm gonna say the next few days, but I'm really saying about a week and a half, so 10 days total, her body's going to go through a training pretty much, right? It is because she's been encoded with the mRNA messenger, and it is right now trying to build itself up to communicate with the entire body to create a defense mechanism, or in this instance, help itself trigger an immune response by teaching its cells to make small, harmless pieces of the virus within the body. Okay? She's going to go through some stuff, but she may not experience any adverse effects until the booster shot comes into play, which actually helps increase the efficacy rate. All right? How do mRNA vaccines work? I kind of explained that in just a little bit, but we're going to be a little bit more specific. The mRNA vaccines for COVID-19 are injected into the muscle of your upper arm. Once the mRNA is inside your body, your muscle cells will flow the genetic instructions that tell them how to make the spike protein. a small harmless piece of the COVID-19 virus. Now remember, the mRNA vaccine does not contain any sample or portion of this. This is a synthetic version of the virus. All right, that's harmless. Okay. Now, the spike protein does not cause infection or cause people to become sick from COVID-19. After the spike protein is made, your immune system will recognize that it doesn't belong there. Now, as a result, your immune system will begin making the T cells and antibodies that recognize the spike protein. This is that journey that I was talking about over 10 days. Now, this process teaches your body how to recognize and destroy the virus if you ever infected with COVID-19 in the future. So it's a beautiful way to do this, and it's a safe way to do this because we're not using a dead or a copy of it. I mean, you know. All right, so let's get into this real quick. Let me see if it'll play. And it's just going to show you how this experience happens within the body. This is from Science Photos, so shout out to Science Photo Library. So this is what happened to CK a couple of days ago. Well, yesterday. All right. There you go. You see it developing the spike proteins, and now it's going all over the body and it's trying to create. It's pretty much the process. Now, it ain't going to look nothing like that, but you know what I'm saying? It's just a 3D visual. I thought that would be effective. All right. So how mRNA COVID-19 vaccines work. All right. So there's a process. Now, mRNA vaccines give the immune system genetic instructions to recognize the COVID-19 virus. You have your spike protein, which is the spike mRNA. All right. So you have the mRNA going into your arm, your upper arm, the fatty side, where the tissue's at. And that's where it needs to go because it needs to latch on to that fatty stuff in your muscle. Now it's going to get into your cell. Remember, it doesn't go no further than the cell membrane. It doesn't get into the nucleus of the cell. All right. It stays outside, close to the ribosomes if they need to be created or whatnot. I'm taking you through just the images, and then I get to the bottom. And then after that, you start to develop the antibodies and build up the T cells. And then next thing you know, you have these antibodies and T cells that have developed to help you fight off the virus. That's just based on the images at the top. Now, at the bottom, it says mRNA vaccines give instructions to the cells in your body to make the spike protein a small harmless piece of the COVID virus, which we have redundantly repeated. The mRNA vaccine will be injected into the muscle of your upper arm, which I explained. Once the vaccine is inside of your body, your cells will follow the instructions that tell them how to make the spike protein. This is the beauty of technology, is that we have the technology now to tell our immune system how to defend itself against RNA vaccines. All right, so the spike protein does not cause people to become ill with COVID-19. Like I said, it's going to be redundant. And then after your muscle cells make the spike protein, your immune system will recognize that it doesn't belong there, and then it'll continue to build the immunity in the T cells. So let's move on from there. Now, how effective are mRNA vaccines? And the Pfizer, BioNTech, and Moderna vaccines are about 95% effective in preventing COVID-19 infections. More data will become available as time goes on, as more people are vaccinated. And this, the beauty of it all, is that we continue to get more data. There was, I was reading an article, they were talking about Moderna could possibly, gain or create immunity for up to 2 years, possibly. But we don't have the data for that. So it's a process. All right. And we're here. So we know that they've been working on this for a year now. So we know there's something that's going on where they have enough data to know that, hey, this thing's going out further than a few months. Because initially they were saying 45 days. Then they went out to 2 months. Then they went out to 3 months. Then they went out to 4 months. It keeps increasing. After the booster shot, that second shot increases the efficacy, and they're starting to know now that, hey, we can start to measure how much longer this is going to happen because we have it out in the public now. And people who've had 2 doses, we still trialing them. But anyway.
This is how coronavirus invades your body, sinking its crown-like spikes into your cells, using molecular deception to pick their locks, and hijacking your body. But there is one way to prevent this, by using one of the virus's weapons on itself. Hey smart people, Joe here. Right now, there's about 7 billion people all waiting for the same thing, a vaccine that will protect us from the virus causing COVID-19. And if you're like me, you want to know what's in it, what makes it work. Making a vaccine and getting it out to the public is a long process with a lot of steps, so we can make sure the vaccine is safe and effective. And it's pretty typical for that to take 10 years or more. In an emergency, like this pandemic, well, we can't skip any of those steps, but we can speed this up by doing some of them at the same time. But none of that can happen until you figure out the first step. What do you put in your vaccine that will make it protect people? And that's what we're going to talk about today. We're going to visit a lab and meet some scientists who study the coronavirus. We're also going to meet a really big, awesome science machine and learn how they used it to design this, the key ingredient inside the very first COVID-19 vaccines. Now, here's my goal with this video, to show you what exactly is in the new COVID vaccines that makes them work, and how they got made faster than any vaccine in history. My hope is that you'll be better informed when you get your shot, and you'll have a new appreciation for why science like this is so important. This is how to make a COVID-19 vaccine. It turns out that some of the most important research for making the COVID-19 vaccine is happening right down the road from me at the University of Texas, which is pretty cool because I did my PhD right here. And in here is the lab of Dr. Jason McClellan. He studies how pathogens like coronavirus cause disease.
There are 4 human coronaviruses that occur seasonally and generally cause the common cold. And then there have been 3 coronaviruses that have caused pandemics. And that's the first SARS coronavirus back in 2002, the MERS coronavirus in 2012, and now SARS-CoV-2, which emerged earlier this year. At the end of December 2019, it was on the news that there was these pneumonia clusters in China. In the scientific community, we thought maybe a new flu virus or possibly a coronavirus. I was actually snowboarding with my family, and my collaborator, Barney Graham, at the Vaccine Research Center at the NIH called, and he said he's been in contact with US CDC, Chinese CDC. It looks like it's a beta coronavirus, similar to SARS, and they wanted to move rapidly, try and make a vaccine. And I said, we're definitely in.
So you're just like scrolling through your phone in the ski lodge. You're like, we got to get to work.
So while the rest of us were focused on royal family drama and just hearing the first mentions of coronavirus for the very first time, scientists like Jason knew this was serious, and they were already getting to work. As soon as researchers in China decoded the virus's genome and published it online, Jason's lab could start designing a vaccine.
I texted Daniel Rapp, my graduate student, and let him know to be on high alert, because as soon as we get the sequence, we're going to move as quickly as we can.
Jason was on that winter vacation and texted me that it was a coronavirus, and eventually in early January, the sequence was released online publicly. And then things started moving pretty quickly.
Let's step back for a minute. What does a vaccine do? Well, it trains your immune system to know what a germ, like a virus, looks like, so that it can recognize the germ, fight it off, and keep you safe without you getting sick. This is the virus that causes COVID-19. The outer shell is made of a few different kinds of proteins, but these proteins sticking off the side are the most important part, the spike. These spikes are what give this family of viruses their name, the coronaviruses, because they look kind of like a crown. The coronavirus uses that spike to sneak into our cells. The three-dimensional shape of that spike is super important because that exact shape is what lets the virus latch on to receptors on the outside of our cells, almost like picking a lock. And then it sneaks inside. Those shapes sticking out on the outside of a virus are also what your immune system is feeling for, to figure out if this is a foreign invader, if it should attack or not. The problem is, the first time your body sees a virus, your immune system responds so slowly that the virus has time to make gazillions of copies of itself. You can still get very, very sick. That's what's great about a vaccine. It trains your immune system what to look for, so when the real virus shows up, your body can respond super fast and destroy the virus before it has a chance to hijack your cells. So what's actually in a vaccine? Well, sometimes a vaccine has a weakened or dead virus. That's how polio and measles and mumps and some other vaccines work. But these days, a vaccine usually just contains a little piece of the virus. The newest COVID-19 vaccines, they're just the spike. But for that spike to work as a vaccine, to train your immune system to recognize the actual virus, it has to have the same three-dimensional shape as the spike on the whole complete virus. But making the spike all by itself, not attached to the rest of the virus, it turns out to be really hard because the spike is actually pretty floppy, just floating around on its own. It doesn't look much like the spike on the actual virus. And this is the key thing Jason's lab figured out how to make. For years, they'd studied SARS and MERS viruses, which are really closely related to the virus that
All right, so it looked like I should be back. Let me see when I come back, man. All right, I'm gonna wait a second. Told you I'd be back. Yeah, I know we back. Nah, man, I ain't blaming the anti-vaxxers. I kind of figured that they would PBS would have an issue with that particular video because I could have did it in a different way. Like I could have just, you know what I'm saying? I could have downloaded it myself, but in a presentation form, it still shouldn't have popped it, but it's all good. It's public information. But anyway, what he was saying when he was getting into the video was that they had to, Jason had to, or whoever the dude is, had to, he had to build the protein part to help the spike stick. That's basically what he was getting at. Once they figured out how to build a protein part to help the spike stick, then they could be able to use it to fight against COVID-19. So that's basically what took place. They mapped it all out. They got the information. They got into the lab. They had to figure out they figured out how to make the spike. Once they had the spike in play, then they had to figure out how to keep the spike sticky. The spike needed help, so the spike needed a protein. They lasted to a protein which forms the spike that's how you get the spike protein. Once you get the spike protein, you was good to go and do everything else you needed to do. That's pretty much how they made that vaccine. Hold up, I ain't trying to play it right now, but that was cool. But that was pretty much it on that part. It wasn't the whole video. It was only a little bit. So, moving on. I'm gonna share my screen again, and we'll get back to it. Y'all should be able to see my screen again. All right, there we go. Nah, man, we ain't blaming the anti-vaxxers. Although, you know what I'm saying, I have had some reports before, but nah, it was all good. All right, so mRNA vaccine ingredients. Moderna mRNA vaccine ingredients is simple. It's not a lot of information here. It's just simple. The Moderna COVID-19 vaccine contains the following ingredients: the messenger ribonucleic acid, the mRNA, and then you have lipids, which is the SM102, O2, which is the polyethylene glycol, PEG 2000, the mystriol, glycerol, DMG, the cholesterol, and I2, the sterol, SN glycerol 3-phosphochloride, which is the DSPC, tromethamine, I always mess it up, sound like Chinese, tromethamine, if I say that right, and then hydrochloride, and then acetic acid, sodium acetate, and sucrose. Y'all want to look them up? All right, so again, there's pretty much just an image. Y'all know what that image is? See it everywhere. Can it be altered? Pfizer's mRNA vaccine ingredients is similar, but not the same. All right, it includes the mRNA, lipids. This has a 4-hydroxybutyl azendiol, and then it has a bis hexene 6 NDL, and then it has a bis 2 hexyl decanoate, and then it has 2 polyethylene glycol 2000 N, and then it has the N. What is that? Ditetrol classamide, and then I2 disterol SN glycerol 3 choline, and then cholesterol, potassium chloride, monobasic potassium phosphate, sodium chloride, diabasic sodium phosphate, dehydrate, and sucrose. This is what CK got in her body right now. Yeah, she on it right now. All right. So the mRNA vaccine ingredients, there are other vaccines currently on the market that have the exact ingredients. The Moderna mRNA vaccine, which has not yet received EAU, but at the time this was written by Vanderbilt, it has already captured the EAU, but it is based on a similar construct with the PEG 2000 lipid delivery system. At this time, there are no cases of serious allergic reactions associated with the Moderna vaccine in the reported clinical trial data. Polyethylene glycol, PEG 2000, is a component of the vaccine and anaphylaxis. Now, peep this. Severe life-threatening allergic reactions to PEG has been rarely reported with PEG 3350. Bowel preparations and prescription medications such as menthol, prednisolone, whatever you Ace-Tec, or Depomedrol. When the heat said when you need them. Now, if you think you have had anaphylactics with one of these products, you should contact occupational health and your primary care physician. You can consult with our allergy service. So Vanderbilt has a site set up for COVID-19 potential candidates who may have had some kind of serious allergic reaction to the PEG 2000 lipid, or the PEG 3350, which could trigger or cause an allergic reaction, severe allergic reaction. And, they were set up to consult and deal with people on that level to educate them on the vaccine ingredients and look at their shot records and history of other vaccines they may have had. All right, my bad. So moving on. Conclusion. After properly identifying the ingredients in the mRNA vaccines, Pfizer and Moderna, we can honestly conclude that neither vaccine possesses ingredients that are harmful or dangerous to the population. Both vaccines do not contain eggs, preservatives, latex, or stranded or dead virus. Although mRNA vaccines are new copies, companies have been working on this technology for almost 30 years. Each company has made a safe and effective vaccine with a short list of adverse effects and a list of safety measures to prevent misinformation and distrust. We do know mRNA vaccines stay in your body only a matter of a few days before it dissolves itself away, and the second shot primarily is a booster shot. Reports have shown that the vaccine immunity lasts as much as 4 months, with some experts claiming longer. While in the middle of a pandemic, our odds at achieving a rightful solution has just improved with the mRNA vaccine. Again, all sources are in the description of this particular YouTube video. I'm actually going to put the video link in the description for y'all, or actually in the chat for anybody interested in watching that further. So that was pretty much a brief little lesson on what mRNA vaccines are with the stoppage of this particular video, and I apologize. Bear with me one moment. I'm gonna get that and put that in the chat for y'all. And yeah, CK, we know they've been raking shop on that, but some people, yeah, you're right. The monetized people, they care about their little stuff. I done got them a couple views. I'm gonna give them a few more. All right, before I move on, man, anybody got any other question regarding the mRNA vaccines? Peace, brother Lima, sister Juju, can't get right. I see y'all just got on here. Everything is everything. Number one troll, always asking the wrong questions. CK thought I got sniped, but we came back. I can edit the video and attach it if I feel like it. Bruce, Ronald telling everybody, what up, Jerome, LB, Robert Rand, Charles was good, Morpheus, Benita, Chavis is in the building, DC, from West Africa, what's good, Zane, Lamar, I seen Vasa, I ain't seen him since. I seen Sutek as well. Black African Power, what up, Rasta man? But does anybody have any questions regarding on what mRNA vaccines are? Try to hit it to where we can actually know exactly what it is, what the ingredients are, how it works. Just to keep it simple. Yeah. Okay, so I want to go ahead and since we don't have any questions, I want to get into some latest news, man. Some latest news. Let's deal with the media real quick. Again, everything that I show you or share on the screen will be in the description of the video. All right, so I just wanted to go ahead and deal with political fact and get into a claim that was on Facebook. It says a new coronavirus vaccine contains toxic ingredients and are more dangerous than getting COVID-19. That was a claim posted December 15, 2020. No, the new coronavirus vaccines are not more dangerous than COVID-19. So we have people that are afraid of getting the actual vaccination saying they rather take their chances with getting COVID-19. I just want to let y'all know what you're up against. Whether you experience small symptoms, mild symptoms, or severe symptoms, you're going to experience some type of symptoms. Now, the longevity of those symptoms are at play because further on down the line, you could have some serious lifetime lingering illnesses that could occur from SARS-CoV-2, COVID-19. All right. So it says if your time is short, they cut straight to the chase. The ingredient list of the vaccine does not include any of the toxic contents listed in the post. So whoever put the post up, and we'll get into it, says the leading vaccines, which have the best test for several months and thousands of people, can cause mild or moderate short-term reactions resolved without complication. They are not more dangerous than the virus, which has killed over 300,000. Now 400 plus thousand, almost 450,000 people now. All right, so let's go ahead and move on. So a lengthy and popular Facebook post attributed to an alternate medical doctor makes several misleading claims about the new coronavirus. Among them, that the new mRNA vaccines can alter human DNA, which we already dealt with previously, that we don't need a vaccine because we've reached significant herd immunity, which we haven't, and that anyone who has COVID-19 would not benefit from the vaccine, which is also not true. The post also erroneously claims that the vaccine contains toxic ingredients like aluminum, mercury, and possibly formaldehyde, and then concludes that ultimately receiving the shot is more harmful than contracting the disease itself. From just a brief lesson that we actually dealt with on the vaccine, we already know that that's not true. All right, it says, here's my bottom line. I would rather get a COVID infection than get a COVID vaccine, reads the post, which accompanies and imagines a man wearing a white physician's coat that would be safer and more effective. Two problems. The list of ingredients cited in the post appears to be pulled from the air, from thin air. An amount of evidence shows that getting infected with the virus is far more dangerous than vaccines. The vaccines have been studied for months and have been proven to be safe and effective in tens of thousands of people. COVID-19, by contrast, has killed over 1.6 million people around the world, which that number just increased. The post was flagged on, as a part of Facebook effort to combat false news and misinformation on this feed. You can read more about that with the partnership with Facebook. When Pfizer-BioNTech COVID-19 vaccine received the EAU from the FDA, December 11th, its ingredients list was published online in a fact sheet. All right, for each recipients and caregivers. Now, the fact sheet is also in the description for Moderna and Pfizer. That list includes mRNA, lipids, salt, sugar, and saline solution. None of the supposed toxic ingredients listed in the Facebook post appears in the list. The mRNA used in the Pfizer and Moderna vaccines represent a new vaccine technology, but the research behind it has been underway for some time. It's not a novel strategy, said Dr. Paul Afit, doctor of the Vaccine Education Center at Children's Hospital of Philadelphia. That's why Uncle always say y'all can source chops. This particular notion of using messenger RNA in a vaccine has been around for 20 years, actually almost 30. This is just the first product to get above the water to become a commercial project. Traditional vaccines introduce an inactive or weakened version, which we're aware of a virus in the body to fight against. By contrast, Pfizer and Moderna COVID-19 vaccines use messenger RNA, which is a fragment of the virus genetic material, which we're familiar with. Now, other ingredients of vaccines. You can get into the rest of that, but it really doesn't have to deal with what we dealt with because we got into all of that. Okay. Hey, your boy go right there, Rush Limbaugh. The American Medical Association, I thought he was going to hit me with the Aboriginal. Rescinded the previous statement and now says hydroxychloroquine is okay for COVID. That's false. We ain't even got to get into that. We know that's false. We have ruled out hydroxychloroquine. Do not take it. Don't put yourself in position. Well, we smart here. Don't put ourselves in position to take that nonsense. So we really ain't got to deal with that. So I can move on from that. All right. If you need any evidence, if you need to refer back to this for a conversation you're having with a family member, friend, or somebody on Facebook or any other social media site, the link is in the description. All right. So let's go deal with another fact checking opportunity. Boom. If I can get rid of the ad. Nope. All right. So it says fact check. No, the COVID-19 vaccines don't have toxins. All right. This was a little bit later on in December. Okay. And they're talking about the vaccine ingredients for the coronavirus is just plain false. Okay. Now they take into news media stations and all kind of other things to fight these misconceptions. So instead of me talking, I'm gonna listen to the article. Well, we're gonna listen to the article.
Just a quick word from our sponsor.
Oh man.
Is your gut as healthy as it should be?
I can't deal. 7, 6, 5, 4, 3, 2, 1. All right. So it's about to play now.
Fact check. No. The COVID-19 vaccines don't have toxins. A lengthy and popular Facebook post attributed to an alternative medical doctor makes several misleading claims about the new coronavirus vaccines.
Can't get right. Peace to you, brother. I did not mean to skip you. Trust me. Charge it to my head, not my heart.
Among them, that the new mRNA vaccines can alter human DNA, that's pants on fire, that we don't need a vaccine because we've reached significant herd immunity, we haven't, and that anyone who has had COVID-19 would not benefit from the vaccine. Not true. The post also erroneously claims that the vaccine contains toxic ingredients like aluminum, mercury, and possibly formaldehyde, and then concludes that, ultimately, receiving the shot is more harmful than contracting the disease itself. Here's my bottom line. I would much rather get a COVID infection than get a COVID vaccine, reads the post, which accompanies an image of a man wearing a white physician's coat. That would be safer and more effective. 2 problems. The list of ingredients cited in the post appears to be pulled from thin air, and amount of evidence shows that getting infected with the virus is far more dangerous than the vaccines. The vaccines have been studied for months and have been proven to be safe and effective in tens of thousands of people. COVID-19, by contrast, has killed over 1.6 million people around the world. The post was flagged as part of Facebook's efforts to combat false news and misinformation on its news feed. When the Pfizer-BioNTech COVID-19 vaccine received emergency use authorization from the U.S. Food and Drug Administration on December 11th, its ingredient list was published online in a fact sheet for recipients and caregivers. The list includes mRNA, lipids, salts, sugar, and saline solution. None of the supposedly toxic ingredients listed in the Facebook post appears in the list. The mRNA used in the Pfizer and Moderna vaccines represent a new vaccine technology, but the research behind it has been underway for some time. It's not a novel strategy, said Dr. Paul Offit, director of the Vaccine Education Center at Children's Hospital of Philadelphia. This particular notion of using messenger RNA in a vaccine has been around for 20 years. This is just the first product to get above the water to become a commercial project. Traditional vaccines introduce an inactivated or weakened version of a virus for the body to fight against. By contrast, Pfizer and Moderna's COVID-19 vaccines use messenger RNA, which is a fragment of a virus genetic material. It works by giving the body instructions to produce copies of the protein present on the surface of the coronavirus without causing sickness from the disease. The immune system then learns to recognize the protein and produce antibodies against it. Other ingredients of the vaccines. As for the rest of the ingredients, the lipids are nanoparticles used to encase the RNA. The salts help keep the pH, or acidity, of the vaccine close to that of a person's body, and the sugar safeguards the nanoparticles when they're frozen and stops them from sticking together, according to MIT's Technology Review, which spoke with experts to help decode the contents. Before injection, the vaccine is mixed with the saline solution, just as many intravenously delivered medicines are, the report said. Moderna also released a similar list of ingredients through the FDA, with a slight difference that may explain the different storage needs for each. The Pfizer vaccine needs to be kept at -70 degrees Celsius, while the Moderna vaccine can be shipped at -20 degrees Celsius and can be stored in a refrigerator for up to 30 days after that. K. Viswanath, a professor of health communication at Harvard School of Public Health, told PolitiFact that the post uses common anti-vaccine tactics, such as using what he calls scientific language, as he calls it, and claiming it comes from someone credible. This kind of tactic to list ingredients that may or may not be there is not new. Anti-vaccine groups have always used a list of ingredients even after ingredients have been eliminated from vaccines, Viswanath said. They misinterpret and distort scientific data to advance their agenda. Some people who get vaccinated may develop mild or moderate symptoms as their immune system responds, including headache, soreness, or fever, but these side effects are short-lived and not considered serious or life-threatening. Some skeptics of the vaccine have pointed to the high survival rates for some groups of people who contract COVID-19. But the vaccine isn't intended just to prevent deaths, health experts note. It's also about preventing the spread of the virus and infections that could lead to significant long-term health issues. Rigorous safety review. While the vaccines were developed in record time, they went through rigorous review processes before they were deemed suitable for public use. For example, the vaccine developed by Pfizer-BioNTech has been studied in clinical trials involving about 44,000 people, with half receiving the vaccine and the other half receiving a placebo. To receive emergency use authorization from the FDA, the manufacturers had to follow at least half of the participants for at least 2 months after their vaccinations, and the vaccine had to be proven safe and effective in that population. An FDA analysis of that vaccine's data found no specific safety concerns that would rule out using it for people aged 16 and older. For Moderna, 30,000 participants have been studied in its trial, with half receiving the vaccine and half receiving a placebo. An interim review of the data found that the vaccine was safe and well tolerated, with an efficacy rate of 94.5%. Akiko Iwasaki, an immunologist at Yale University, called the post irresponsible. Where's the data? How can they claim that the vaccines are contaminated with these agents? Iwasaki wrote in an email, "Getting the vaccine is absolutely not more dangerous than contracting the disease. The safety of both Pfizer and Moderna mRNA vaccines have been rigorously tested with no serious concerns. COVID-19 has a case fatality rate of 2.3%. Vaccine 0. Addressing some of the post's other claims, the U.S. has not reached herd immunity that would significantly hinder the spread of the disease, as the Post suggests. In a recent interview with NPR, Dr. Anthony Fauci, the director of the National Institute of Allergy and Infectious Diseases and a member of the White House Coronavirus Task Force, predicted the country could begin to achieve early stages of herd immunity by late spring or summer if people get vaccinated. I would say 50% would have to get vaccinated before you start to see an impact, Fauci told the outlet. But I would say 75% to 85% would have to get vaccinated if you want to have that blanket of herd immunity. People not needing to get vaccinated because they already had COVID-19 also doesn't wash. There isn't enough information yet to say if or for how long someone is protected after they recover from the disease. Some early evidence suggests that natural immunity may not last very long, but more studies are still needed. The vaccine is expected to provide stronger and likely longer-lasting immunity. In its recently released vaccine recommendations, the Centers for Disease Control and Prevention said that vaccination should be offered regardless of whether a person has had a prior infection. PolitiFact ruling, a Facebook post that makes several unsubstantiated claims about the new coronavirus vaccines, says the shots contain toxic ingredients and are more dangerous than getting COVID-19. This is bogus. The ingredient lists for the vaccines do not include any of the toxic contents listed in the post, and the leading vaccines, which have been tested for several months in thousands of people, are not more dangerous than the virus, which has killed over 300,000 Americans and more than 1.6 million people worldwide. Pants on fire.
Next article.
All right, so real quick, I want you to put, I gotta put this in there. Because you have people who like to throw out a stat, right? They throw this stat out talking about 99% of the people recover from COVID, which is really disrespectful and disingenuous to say that. Because when you put things into perspective, the fatality rate at 2.3%, the vaccine is 0%. Which one would you rather have? You're looking at it from a global perspective. You're talking about millions of people dying from a virus, and that's supposed to be, well, everybody else recovering. Fool, do you not realize that that pushed one of your family members or 2 or 3 of your family members in jeopardy? Especially if they have underlying conditions and have a high risk of actually being taken out from this thing. And like I said, developing symptoms over time. Like we gotta be, we gotta do a better job with that. But I think this quote right here, the safety of both Pfizer and Moderna mRNA vaccines have been rigorously tested with no serious concerns. COVID-19 has a case fatality rate of 2.3%. Vaccines 0. All right, so let me move on to this other part that I wanted to kind of talk about real quick, man. Then I'm gonna wrap up. I ain't gonna try to strain this out. I'm gonna try to go in there and edit and fix the video. But let me see. Let's get to it. So Pfizer and BioNTech vaccine appears to work against the new COVID variants. Study finds this research U.S. records daily is day. All right, so new research. All right, from the University of Texas, U.S. drug maker Pfizer suggests that the company's COVID-19 vaccine developed with Germany's BioNTech is still effective against the new highly contagious variant. All right, so here's the actual article for it. I did not put this in the description. I will put it in the description of the video, but I'm going to put it into the chat and make sure that the chat actually has this particular article. Now, without reading this particular article any further, we already know that they're doing testing on the strands, and we know that it's more of, it's, how I want to say this. there's a potential new variant, not strand. I said strand, and I apologize. But there's potentially a new U.S. variant that could emerge or has emerged. It's a possibility that a new U.S. variant exists, as it is, popping up in all these other countries. A new U.S. variant could exist. But we shouldn't fret. We shouldn't fear. The vaccine is here. But anyway, in this 6-page PDF document, it has a neutralization of the N501Y, which is the variant that's in discussion right now. All right. So I'm not going to get too much in depth with this particular information, but we all know that the UK and South Africa have been diagnosed with a new variant, right? And long story short, they already been in process with vaccinating people who have been exposed to the new variant. I mean, not really vaccinating people, but you know what I'm saying? Like exposing people to the new variant, pretty much so. Seeing what the data is showing and saying, right? And it is showing and proven to be effective against the new variant. Case closed. In this 6-page article, you actually can read more about it, and they're going to continue to do research and develop more data. Moderna also, they both came out and said this a while ago. So this wasn't really like breaking news, but they're still building on their data, and they're just being transparent. We usually don't get information like this until on down the road. So every time we get a chance to be not ahead of the curve, in most cases, but on schedule with the scientific community, I feel like that's a big responsibility for us as Black people to be on the same page with the scientific community. Yeah, I already mentioned the one in Africa, Robert ran. South Africa, of course, specifically. But, and then whatever the Nigeria thing going on. But anyway, variants happen, they occur. Remember now, about a week or so ago, we did something on the variants, probably 2 weeks ago, when we got into 12 and we started dealing with the 12 team, and discussing variants. And Brother Vincent, Professor Vincent, was not, see, here you go right there, was not fazed or moved by these variants. Wasn't phased or moved by these variants. By the way, he's getting ready to start a new virology class. He's prepping up, and I think the class starts next week. And I actually reached out to him to see if we could actually have him on the Science with Shawn, or possibly the pseudo killers, to discuss viruses and how they behave, and kind of give us some little insight. But with class presuming, I am almost positive that we won't get to hear from him. So if we do, then there's still hope for that, but I ain't hanging my head on hearing from the buddy. You know what I'm saying? I don't know, because he's got shows to do too. He's doing 12 a few times a week, and that's his platform. So it would be nice to have him on so people can ask questions and build and learn more about viruses. But it is what it is. So there y'all go with that data. I actually gave y'all that data there. I don't want to get into this whole article. I pretty much just wanted to give y'all what you really needed, which was the research out of this article, which was more appropriate than anything else. Okay? Because we know about everything else that's going around. We know that there's, deaths and all of those things. And we don't really have to get the CDC report or, any more Forbes information or anything else. We got everything that we need from the article that we actually can source. So we don't have to hang our head on a secondary source. In this instance, regarding the data, we can just go directly to the research and comprehend the research. All right. So Pfizer is stepping up first and saying, well, Moderna has stepped up both and said, hey, look, we cool against the new variant. All right. me see. Let me stop sharing real quick. Yeah, he busy. I mean, I can't hang my hat on somebody that, you know what I'm saying? It was a blessing to get the doctor that we had on the pseudo killers, you know what I'm saying? Shout out to Truth Ministries. He made that possible. And then him and Unc, you know what I'm saying? They kind of took the lead on that, trying to make sure that, you know, came to fruition. And y'all got to see what she had going on. I'm looking in the chat for any questions, man. I know a couple people just now joined, and I'm not going to exaggerate this. Hold on. Not exaggerate, but I'm not going to exhaust the stream. I mean, I can go back to playing. I can go back to playing the conversation for a little bit. Give y'all a little bit, 15 more minutes of that, so we can hit it right at the hour and a half mark, or what's going on? I do that. All right, I'll do that.
Do those have some sort of phenotypic importance? I mean, is there something, like you mentioned, American Indians, do they have some gene that's just been hanging out for generations and then pops up someday when conditions change, or what's the importance of a founder effect?
Right. So the importance of the founder effect is, like you say, it can drastically change the genetic makeup of a population purely by chance. For example, there might be certain MHC, you know, the histocompatibility loci. There might be different frequencies of MHC genes or other metabolic genes or any sorts of genes with various functions, or genetic variants in those genes, I should say, that will be different in different populations, and then those will get carried down. And then I think, as you're suggesting, there might be times when that variant might be more or less advantageous when presented with a new situation. And so it's a way that the genetics can be very complex over time in figuring out what is actually adaptive, what's being selected or selected against, versus what is just kind of there from just a factor of chance in the evolutionary history, whether it's in people or whether it's in viruses.
What about a genetic bottleneck? What's that?
So bottlenecks are somewhat similar to founder effects in that a bottleneck is when you get a restriction in genetic diversity. The most common one, as a virologist, we think about a lot is during transmission, where a single individual might have hundreds of different influenza viruses or hundreds of different HIV viruses within them. They're genetically different. However, during transmission, there's this bottleneck, just like pouring out marbles through a bottle, that only some of them will actually start the infection in the new person. And so there's been a dramatic reduction in genetic diversity during that transmission event, which is very much like a founder effect.
Before we get into the specific variants of SARS-CoV-2, could we have you give us a couple more definitions?
Sure.
Could you tell us the difference between mutations, variants, and strains?
Yeah, so this is a very important point, I think, as we'll get to. And you probably can tell I've been trying to keep them separate as we've been talking, but I conflate them myself. So strictly speaking, a mutation refers to a single change in the genetic sequence. And so you could talk about a single change in a base or a nucleotide, or you could talk about a single amino acid change. So something that changes, the coding of the protein, the coding potential of the protein, or the amino acid encoded in a protein. So that would be a mutation. So if you have a SARS-CoV genome that's 30,000 bases, you might have 3 mutations relative to some other virus, or 3 amino acid changes. So that single genome has 3 mutations. A variant is a pretty loose term. A variant is something that varies. So it's something that is different. And so you could say that the genome that has 3 mutations relative to the other one is a variant. It's a single variant. Sometimes people will talk about variants as talking the same thing as mutations, but as we'll probably get to, some variants have multiple mutations. Then there's a strain, which is another term that's used quite loosely. And that's okay unless you want to get highly specific about it. But strictly speaking, a strain refers to a variant that has a phenotypic difference. So something that is different in some way, so that can be it's better at escaping antibody, or it's better at transmitting, or it maybe causes more serious disease. So some way that it's actually different, not just based on its sequence, because you could have viruses that are genetic variants or similar mutations, but actually don't behave any differently.
Why don't we get into some of the known variants of the SARS-CoV-2 virus? Some of them are quite worrisome, but starting off with Spike D614G. What is that?
Sure. So this is one of the variants that got a lot of attention early on in the spring and early summer during the pandemic. And this was a mutation, D614G, which is an aspartic acid D to glycine G at position 614. So that's the nomenclature there. And spike, of course, is the main surface protein of the virus. It's what binds the receptor. And it also is the dominant protein targeted by antibodies. And so it's what's in the vaccine is the gene for spike, because that is what triggers what we think is a protective antibody response. So this variant emerged, and so it's a mutation, D614G, but it's now in many different variants because there are other mutations along with it and other different variants around the world. And so it was observed in early March to have arisen in multiple countries and quite quickly. And so this was concerning because it looked like you were having something new taking over in multiple places, which in evolution, when you see something happen multiple times, that's a strong clue that it might be conferring an advantage for the virus, that it's natural selection at play. However, later there was data that that mutant or variant had actually been in China in January. And so that led some people to be concerned that maybe it wasn't being selected, that maybe this was a founder effect, that somehow that variant was just overrepresented in the viruses that made it out of China. And so that would be a founder effect that happened multiple times, that it had a head start over the other variants, and that the fact that it eventually dominated was just due to these chance events. However, subsequent work, which has taken a large amount in many institutes across the world, has suggested that it actually might have a small competitive advantage, that this mutation appears to have spread more quickly in various countries when controlling for this founder effect. And so there's complicated models that will do that. And then the other piece of evidence is people have been able to do animal studies as well as studies of the virus in cell culture, which are complementary and also seem to suggest that it is able to spread better than the non-D614 virus, the D614G virus, the D virus. Putting it all together, it looks like it probably does have some sort of advantage over the ancestral SARS-CoV-2 as we know it. And so there may have been some element of founder effects, but there also appears to be evidence for some advantage in terms of its ability to transmit or spread.
Does anybody know what that mutation does to enhance that ability to spread?
It's thought that it binds or utilizes the ACE2 receptor better, that it's able to somehow get into cells better than its ancestral virus, the D virus. So the G virus is it has to do with the interaction and the way it utilizes the receptor, is at least the current thinking. Clearly there's more work to be done in this area, but it also appears to, maybe because of that, grow better in cells. And there is some data that it'll grow better within a person or within an animal model, such that it can perhaps copy more of itself over time. And then you could see how that might make it better at transmitting or spreading, because if someone has more virus in them, they may be shedding more, and so that may make them more infectious to others or contagious to others.
Another mutation of concern is spike N453Y.
Sure.
And MINC.
Yeah.
What's that all about?
Yeah. So in the spring and into June, there were reports of outbreaks in the Netherlands and then later in Denmark on mink farms. So something I knew very little about until this story. But you know,
All right, I'm gonna clip him right there because he about to start talking about some stuff we already addressed on the show regarding minks and things of that nature. The good God, that conversation of that podcast is like a week old. I just been sitting on it for a while. I shared it with a couple of other people, but we've been sitting on it for a little while as far as that data. But when he started talking about minks, that just lets you know. Yeah, we've already covered and had conversations about minks. Easy bands, yeah. You know, we need to be more concerned with new strands and variants because all viruses mutate. All viruses mutate. And understanding that, you know what I'm saying, we kind of deal with it from there. I don't know if anybody watched 12 the other day, but you know, the 12 bill, they were talking about. Hold on, they were talking about dosage, like how much of the vaccine should we be receiving? Does it matter? You know what I'm saying? Does it matter? And there was some good takes and going on, and you know, I do want to play part of that conversation because the UK made a decision on a dose, but I want to play something at the 13-minute mark. And give me a second, and I'm gonna get into that. Yeah, and then I'm gonna let y'all out, man. I wasn't trying to be on here that long. Let me go to 12:55.
The FDA yesterday said it's a bad idea to just do one shot. You know, we have data that says we need to, and there's a good statement by the FDA, which has come out saying it's not a good idea.
That's a really good statement.
Go ahead,
Brian.
Yeah, I was just going to say, I do think it's a little bit less moot just because there is still quite a bit of discussion about this, and this is occurring in other countries. This is occurring in the UK. And, you know, there still are some very strong proponents of it here. I would be a very strong opponent. I have pretty strong feelings about this one. And I don't think it's a good idea. Or at least I don't think it's a good idea based on the data that we currently have. So I don't think it's quite as much a total moot point, but I do really like the FDA statement, and I'm pleased that they pushed back.
Well, what I look forward to here is an informed discussion so that the listeners can understand what the issues really are, and myself included.
All right. So the new stuff has been added since this morning. So there's the FDA statement, and then also SLUI and Warp Speed have said things. So let's put that at the end after we just chat about the science a bit, okay? And we'll come back to that. So Michael Minna on December 18th in an op-ed said we might be able to do one dose. And his statement was based on the Moderna data, which came out, I think came out about that time, but I forgot the timing. But I was only aware of the Pfizer data. And I went back to the two documents that these two companies submitted to the FDA, and there's one table in each one about this. And in the Pfizer one, their estimated vaccine efficacy against confirmed COVID-1 after dose 1 was 82%. I'm sorry, was 52.4%. All right. But it was only a few individuals. So, in fact, I think I have the table there, right? Isn't that it? Yeah.
No, you don't have the table. Where?
The table is for Moderna.
Moderna table is further down.
Oh.
So there are two tables I put in here. They're both from the Moderna report.
Right.
Yeah. Okay. And the Pfizer actually, I just put the percentage, the vaccine efficacy from Pfizer. So estimated vaccine efficacy, 52.4% against confirmed COVID occurring after dose 1 but before dose 2. All right. And the confidence interval was 29 to 68%. Big range there because there are not a lot of people involved in that.
So what's the first part of this statement? I was confused by this, Vincent, because the first part of the statement on the Google Doc reads, says after dose 1 was 82%.
Yeah. I think that's anything after dose 1, which might include dose 2, right? So if you average dose 1 and dose 2, right?
Okay.
But just dose 1 only is 52%.
Dose 1 only is 52%. Okay.
And that's why I said originally Minna's idea was not a good one. But then I don't know if the Pfizer data the Moderna data had come out or not at that time. But now we have them. It's in the report for the FDA, and I probably overlooked it, so my error. But we have table 15, vaccine efficacy of the Moderna's vaccine to prevent COVID-19 from dose 1 in patients who only receive 1 dose. Okay, so only 1 dose. And they're split into 2 groups. You have dose 1, 1 to 14 days after dose 1, and then more than 14 days after dose 1. And in the one, so you get your first dose, and then 1 to 14 days after that, some people got confirmed COVID, obviously, and the numbers are pretty small. In the vaccine group, 5 out of 1,000, and in the placebo group, 11 out of 1,000, which is a vaccine efficacy of 50%. So 14 days is clearly not enough time. However, if you look more than 14 days after dose 1, then 2 out of 983 people in the vaccine group had confirmed COVID, and 28 out of 1,059 in the placebo group, which is a vaccine efficacy of 92%. And that, I think, is where Minna and many other people who have commented on that said, hey, it looks good after dose 1, right?
Still very small numbers.
Yeah. I mean, it's 2,000 here, which is more than the Pfizer, but yes.
But it's only 30 cases, okay? And as I recall, that they didn't want to make a call on this initially until they had at least twice that many and more like 3 or 4 times that many.
Yeah, considerably more than that. I think it was in the hundreds that they wanted.
So I would call that, promising, but...
Yeah,
exactly. That, I can certainly see the argument, but I'm not sure that we have the data to support it at this point.
Yeah, I think you would need more data. I mean, part of the issue is, as you all know, a lot of vaccine is being distributed and not even put into any arm, right? They're sitting in places because of logistics. So it's not clear to me that this would solve that problem at all, right?
Yeah.
We know nothing about the longevity after 1 versus 2, right?
Right, exactly. So to me, there are sort of 2 pieces to this. One of them is that they've looked, after dose 1 for a relatively short period of time in a smaller number of people. And so we don't know anything about longevity, and we don't know whether this would require people to get vaccinated perhaps more frequently than if they had actually gotten the originally designed dose. The other thing that sort of concerns me that's on that sort of idea is that if you look at the original data that have been published for both Pfizer and Moderna in some of their phase 1 and phase 2 studies, they showed antibody titers after both the prime and the boost. And there was a very dramatic difference in antibody titer after that boost. The boost did quite a lot. There is a document, a link that I put in here that I guess we'll talk about later, that the UK used for making this decision about having 1 dose of the AstraZeneca vaccine. And again, I look at the data, and they have neutralizing antibody data after 1 dose and after 2 doses, and they are dramatically different. And so I sort of, sure, we don't know the correlates of protection. And so maybe that lower level of neutralizing antibodies is sufficient. But based on the difference that I've seen from that boost, and based on sort of the low numbers and smaller amount of time with the data that we have here from Moderna, I'm not sure that we need to change recommendations quite yet.
So one of the things that is in this article that I put in this morning is that as Dr. Mascola of the NIH Vaccine Research Center says, we've always known that the amount of vaccine has been an issue. And if you think back, Tony Fauci and others were saying, it might not be till next fall or toward the end of this year, 2021, that we're in a position where we've got a majority of people vaccinated. And this is all the more reason why multiple vaccine platforms are going to be needed, because just the manufacturing capability is tremendous. And this article also reminds us that the Johnson & Johnson vaccine only requires one dose, but we haven't seen the efficacy data and they haven't applied for an EUA or anything yet. And also, regarding evaluation of the existing data from the phase 3 trial, it was always planned, evidently, for Moderna and the NIH Vaccine Research Center to go back and look at that data and evaluate it, and also look at some of the phase 2 data. And so this article spells out sort of the things that would need to be done to provide the FDA with additional data to perhaps go forward with just one dose. So look back at the patients from phase 2, or conduct a new trial to demonstrate that those that got the 50 microgram dose developed a threshold immune response. So this is talking about a 50 microgram dose versus a 100 microgram dose. So this is a different way of stretching the amount of vaccine that's needed, for example.
Yeah. So I have a question, and I don't know the answer, but as you know, when a new therapy is approved and then you want to test something else that you think might be better, you cannot withhold that approved therapy, right? It's called standard of care. So is it an EUA considered standard of care? Because one, it is not.
I don't think so. Because
then if you're under EUA, then you could say, okay, we're going to have a placebo group that gets nothing, even though we know that we have 2 other vaccines that seem to be work. So an EUA does not fall under standard of care. Because that was my question. Right?
I don't know, but I don't see anything that has indicated that that would be the case. I mean, I think these articles would point out that that would be a complication before the other approval of the biologics license application, the BLA.
So I would like to hear a wrap from Brianne on what a boost is.
Okay.
I want to know what it does, and I think I know enough now so that I can understand what you say. Not that I couldn't before. Understand better. I want to know what it does, and I'm interested also in scheduling. If you look at the childhood vaccination schedule, for in the U.S., the timing of the boost is variable depending on the vaccine, put it that way. So what does a boost do? What do you know about timing?
Sure. So what does a boost do is actually a little bit easier of a question. So when you have a B or T cell, first see its antigen, it will make a type of response that's known as a primary response. So it will go from being naive to start to undergo a program where it gets better at its functions, it proliferates and changes into basically something called an effector cell. And eventually, most of those effector cells go away. They undergo apoptosis. It's a good thing. You don't want to turn into a walking lymph node with all of the T cells you had from your first infection ever. Then you wouldn't have any room for the T cells responding to your second infection. And you are left with a group of cells that are memory cells. Those cells are better able to respond when they see the antigen. So they respond both quantitatively better. They divide faster, and they also act qualitatively better. So they can do a lot more. And each time you see the antigen, you sort of get an improvement in those memory cells. So you can get a secondary response the second time and a tertiary response the third time. And each time those memory cells will be getting better at what they are doing, and will be sort of increasing to get to, maybe some level that might be the threshold for protection. So the boost is giving you that exposure for your memory cells in order to allow them to have a second round of seeing the antigen to help those cells differentiate into good memory cells, a lot of them, memory cells that are potentially making better antibodies in the case of B cells, things like that.
So is the improvement just in affinity maturation? So the you have a better antibody, or is there an effect also on, say, the size of the pool of the memory cells, or how readily they respond?
All of the above.
Okay.
So with the B cells, you will see changes in the affinity, so the affinity maturation. You will partially also see some class switch, at least as the response goes on. It doesn't necessarily have to be a memory cell. There are more cells than there were before the response, and those cells have changed their ability to respond.
So how do I decide when I'm going to do a boost? And can I wait too long?
So I have looked into a lot of this. And if you look in Plotkin's vaccines textbook, which lays out some of this quite well, it is very clearly stated that 3 weeks after the primary inoculation, the primary response has reduced enough so that you have a boost. So the idea with a boost is you need to wait until the primary response is done. So you don't just have the effector cells get rid of your boost. You actually need to be working with the memory cells. So
at least 3 weeks.
At least 3 weeks is that definition, yes.
So how about the other end?
So I have never seen a question of too long. And I will say that, the experiments I did as a graduate student, I gave my boost to 8 weeks, always. But in the situation that we are currently in, we want to get you into that boosted state as early as possible. And so you can see that's why Pfizer is going at that first earliest time point, 3 weeks, in order to get you. Yeah.
The reason I ask is that now this is just hypothetical. I'm just talking. I'm not advocating anything. But if one were to, say, alter the distribution protocol, one potential consequence of that is that people get their first shot and then get boosted instead of at 3 weeks or 4 weeks, say 2 or 3 months down the road, or even longer, 6 months. And my question is, in terms of the effectiveness of the boost, I'm not talking about whether or not the primary vaccination would be effective, but in terms of the effectiveness of the boost, is there a downside to that? I don't know the answer to that.
I think that there probably is a sweet spot. And I think that sweet spot is probably going to vary from vaccine to vaccine. As you mentioned very well, the currently approved vaccines have different amounts of times for that boost. I could certainly imagine something a little later being good, but I just would want to see the data on that. Because I think that there probably, like I said, is a sweet spot. There could be a place where it is a bit too long, and there could be a place where it seems to work quite well.
And in terms of these vaccines, we probably don't know. We don't know what that sweet spot is. They've waited at least 3 or 4 weeks, so they know they're going to get a boost. And probably the strategy is to make it quick enough so that you get everybody boosted to maximum potential as soon as possible.
Right. That is my thinking as well. They may have some of that information done animal studies that they haven't published, but we haven't seen any data on that.
Great. Thank you.
All right. So, Chavis, she teaches, right? So she's talking about a biology book, but I'm gonna go back to, because I know it's a delay. Let me go back a minute. That's why I wanted to hear her speak about this. That's why I sped this process up, because this is what she teaches. This is what she do.
One potential consequence of that.
It is very clearly stated that 3 weeks after the primary inoculation, the primary response has reduced enough so that you have a boost. So the idea with a boost is you need to wait until the primary response is done. So you don't just have the effector cells get rid of your boost. You actually need to be working with the memory cells. So
at least 3 weeks.
At least 3 weeks is that definition, yes.
So how about the other end?
So I have never seen a question of too long, and I will.
Yep, that's it. I ain't got to play it no more. Yeah, it's you got it. But yeah, man, I really wanted to play what Mina had to say, because she's really, 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 really wise about administering vaccines. She's teaching people how to do this. You know what I'm saying? I mean, not Mina, but Brienne, discussing what Mina was saying and how to do the doses and all of that. But you know, a lot of this data we get, so we cool on that end. I'll put this in the chat for anybody interested in actually, you know, trying to watch the whole thing through. Yeah, I know you'll find a book. It's yeah, she said it. But that's that. So hour 45 we've been on here. I want to support Shawn, just not like that. Yep. I know, he know too. Since when did the number one troll not go over there? That's your buddy. Y'all talk on the phone. Remember, he was all in the chat. Call me back. But questions. The man and try to smash. He tried to smash the troll, y'all. Let me quit. Let me stop. So again, Racanelli is starting the class next week. And Brienne starts class next week. I'm trying to see something. Let's see what they got going on. I hate to do this live while y'all right here, while I try to be like nerdy. Spike protein, E protein, M protein, RNA protein. Look who it is, y'all. This is interesting. Course details, and then you can get the premium. So yeah, I'm gonna get this to y'all right now. CK, put this in the pseudo killers. There you go. Oh, if Unc on there right now, man, you know what I'm saying? Then go join if he on there right now. I'm not trying to hold nobody up. You feel me? But what I just put in there, look, COVID-19 fundamentals. Here go to class. Y'all know who Racanelli is. You can, there you go. All the way through. There he go right there as a part of it. You know what I'm saying? You got all these videos right here. Take the quiz. Get your articles. I don't know what you're gonna need them for. But you are currently in a preview mode. You have to create a free account to save your progress and access more questions. So we can go on here, create a account. This one just say free though. I don't know about these other ones. Let me see. Okay.
COVID-19 complications.
Okay. But in order for me to take the quiz, I gotta sign in and register and all that. All right, so I'm gonna be a nerd, man. I ain't gonna hold y'all up. Yo, Shawn, did you say Black Panther Brother Ben's class start next week? Yeah, if you go to Brother Ben page, man, he doing they call it, well, we call it Kimoyo, but you may want to call it African spirituality. I don't like to use that word at all, but Kimoyo. And he's doing that class. Yeah, he's going to get ready soon. He's got a bunch of students that signed up for it, so we're going to deliver on that and you know, get ready to enjoy the journey. What was the name of the textbook that was mentioned? Jerome, Brother Chavis, put it in the chat. It is called Deploykin Vaccines. Chavis, put the name of the book in there. Chavez. Google Scholar said they will block you if you disagree. Oh, yeah. If you go over there on that platform, they definitely gonna block you. But hey, look, man, I just gave y'all a very useful link, man. You know what I'm saying? Just to get some basic knowledge. And y'all seen me doing this online, but you know, this make us better right here. Like this, you know, this college level information that we actually getting. And I just went to the fundamentals and we seen it that is him. So now we actually have the resources there, and I gave y'all the little link. They even let me do this a little bit more. I mean, I don't know how serious y'all are about like learning certain stuff, but has anyone died from any vaccine in history? Nah, can't get right. Nah, bro. Ain't nobody died from no vaccines. Vaccines is save more lives than somebody can claim. Remember that people have died from being administered a vaccine because of human error. Now that has happened. Human error has caused deaths, not a vaccine. We saw that in Samoa incident where the nurse made up a bad batch of a vaccine, administered it to 2 kids, and they died. You know, we read that on pseudo killers when I was arguing my point against old boy regarding him saying the polio vaccine. But you know, when you read it, you know what I'm saying? That information came out. But look, if you ain't got no mask on, so I, you know, just going down the little website, right? Just being nosy, like doing what we need to do. They even got this updated information right here. So, you know, I like to scroll through and look at stuff, and then when like I'm sitting here and I'm not doing it, and I kind of deal with it. But there are no specific clinical features that can reliably distinguish COVID-19 from the viral respiratory infections such as influenza, SARS, pneumonia, and tuberculosis. Common symptoms, severe cases, complications. What have I always said when I start talking about the complications? Your lungs, your heart, your intestines, your liver, your dang on kidney. And then they get into that. Approximately 1 in 6 people with COVID experience clinical deterioration and develop complications in the second week of illness. Oh, you cool the first week, but that second week you get in your grill. Recovery time is approximately 3 to 6 weeks. This is good information. Period. Look at that. Bacterial resources, COVID-19 complications. Unc always want me to talk about the complications. You know what I'm saying? Get into that. Like, this is not stuff that we haven't already addressed, but this is stuff that's good. This stuff is good. Like, this is a heck of a resource right here. You know what I'm saying? Like, this the dagger. Like, this get busy. This put you all up on game. And I don't know if CK is still in the chat or not, but you need to Discord that. Like, this, people need to get this information. Now you can become a student. You know what I'm saying? You can pay your tuition. You can create an account and get the free stuff. I don't know how much of that course they're gonna give you for free though. They might let you just do what you see there, but there may be some extra work that will be. And look at all the references. How many sources they got on here? 39. Yep. Lancet, New England Journal of Medicine, few doctors. It's a lot of stuff. Look, by the way, if y'all see this, all you got to do is highlight that right there. Copy it, put it in there. You can pull up the sources. The ones that ain't got a direct link, copy it like that. Go get the source for whatever you want to do. That's got to deal with blood clots and TAM receptors and all of that. So that's how you do that. If you can't click on the links to get the sources, that's how you do that. Period. And you can get all 39 of the sources. Same thing with this one. That DOI number right here, you should be able to pull that up if it lets you. Because the DOI is listed in those New England Journal of Medicines. And you can go to the DOI number 10.10656 and /, and then it'll be that New England Journal of Medicine should let you pull. I seen the MUI source. All right, then you just keep on going, just keep building, because all of this is coming up out of the COVID place. See? Table of contents, risk factors. And you know, I just like to deal with the risk factors because people like, I don't want to get no vaccine. Okay, well, this is what you up against, buddy. Oh, there's a video right there. Yep. COVID-19 fundamentals. That's where we just came from, the fundamentals. See? We was in the fundamentals right here, and I clicked on that. Oh, let's go down here. I'm just trying to see something real quick. So if I'm blurring y'all eyes up, I apologize. There you go. See there? Look at that. 53 sources. Right there. So we need to take advantage of that, man. People who see, I know Brother Chavis is serious about it because he signed up for some stuff. So he trying to deal with what he trying to deal with on a scholastic level. So let's deal with this 3D. You can play around. Woo! This is what we're looking at. That's the SARS-CoV-2. All right, so let's look at the spike protein closer. All right, there you go. Spike protein. Y'all can't see this. Look at that. Crazy. Can I blow this up? Let me see if I can blow this up. Tapping my mouse. I know I got an Apple product. Okay, so y'all can see that a little bit better. Question for anyone that was afraid to ask. Can't get right, yes. Okay, what do you say? Has anyone died in the vaccine history? Yes, they have deaths recorded, but useless because of it. Yeah. Well, easy bans. I wouldn't necessarily put it on allergic reactions, bro. Because allergic reactions can cause, yeah, I would say that. What side is that? I put it in the chat, can't get right. I'm gonna put it back into the chat. Hold on. So I'm gonna start over. I was here, I went here, this pulled up. Well, this pulled up. This is an online website, right? You can sign in, you take your courses to become a nurse, RN or MP or MDDO, MBBS, PA, or different institutions, okay? So I went to COVID information and then I went to COVID resources. When I went to COVID resources, this information came up. So I'm gonna put this in the chat just right here and just follow along with me while I'm doing this. Yeah, but a lot of people died off human error. All right, from vaccines. It wasn't necessarily vaccines. So I got to scrolling down here and looking at all this stuff, right? And then I got to getting into, you know, the basics. So COVID-19, you know what I'm saying? Knowledge. So we got here. When I got here, I started scrolling down, you know, reading through stuff, and then I noticed, boom, I seen Racanelli right here. So I clicked on this. When I clicked on this, it turned into a course. There's 12 videos. The duration of the videos is an hour and a half. So if you got an hour and a half to watch a few videos, you got 38 questions to answer, and then there's 8 articles for sources. It says, in this course, you will be provided with an overview of severe acute respiratory syndrome coronavirus 2. These lectures will cover the general characteristics of coronavirus and the origin of the coronavirus disease, COVID-19. In addition, you will be instructed on the epidemiological aspects of diagnostic techniques of COVID-19. So you watch one video. Well, really, I'm gonna tell you what you're gonna have to do right now. You're gonna have to watch all these videos. After you watched all these videos and it shows 100% watched on all the videos, then you can take your you can take your quiz. But you have to create a free account in order to take the quiz. When you take the quiz, of course, look, 5 questions per video. Going on. Watch a video, take a quiz. Watch a video, take a quiz. Or watch all the videos, then take the quiz. Some of this stuff we should be able to answer without even watching the video. Like complications. You know, all these things. But mortality rates and things of that nature. It's a free course, man. It's better than what I can do. So maybe next time we do science with Shawn, man, we come on here and we can get busy with it live. You feel me? Hold on, y'all. This what up, You know what? I heard you supposed to be on the big channel today. I think you should just talk about just the pandemic from its inception. I mean, start with the vaccine debate. Yeah. Throw your clips up there, whatever you need to do, but start from the vaccine debate and just kind of walk through the journey of, 2020 turning. The next thing you know is a pandemic. We're in the middle of the thick of things with a whole virus running rampant. And, talk about all the pseudos, how they claimed the virus was fake, black people couldn't get it, and then just get to teaching. You know what I'm saying? And then I'm sure you're gonna try to make it contentious for you. Oh, yeah. Well, yeah. So while I'm about to end my science with Shawn, but I found a nice little resource for people to get some attainable information on the COVID-19 fundamentals. There's an hour and a half worth of videos you can watch, which is about like 12 videos or whatever. And then it's I sent it to you. And then it's quizzes that if you create an account, it's quizzes that you could take. Yeah, it's a learning process, yo. This is an institution, but they let you sign up and create a free account to get the COVID information. So yeah, that's where we at on that. Yeah. So start with the debate. Talk about the pandemic and walk all the way. You know what I'm saying? When you start talking about the pandemic, you know, you can get into the numbers of the real spill, and then you can get into the data. You start you can get into the data, and then getting into the data, you can start, you know, walk them through the process of the vaccine and talk about when it started. And when all of that, you know what I'm saying? Because they don't know this information. They don't know anything about that. But I'm gonna send you I'm finna send you some information on that right now. On just the what the mRNA vaccine is and all the other stuff. Yeah, I know you know. I know. I'm just saying in case you need to pull from your bag. All right. Yeah. You gotta do that. So just go on there and teach the people, man. You know what I'm saying? They're gonna let them make a mockery and do all that criticizing they want to do in the chat. You just teach the people. Yeah. But yeah. So I'm finna wrap up this Mossy show. The people, man, I got the people. I told them you're gonna be on there. So you know what I'm saying? They already know what's up. They know you're gonna be on there. And you know, yeah. Yeah. Alright, ladies and gentlemen, the books is in. The books are in. Yep. They in. They're here.